Thank you, Ian, for that wonderful introduction and for the opportunity to speak to you all this evening and for the opportunity to be at the Oxford Martin School and to have an Oxford Martin School in existence to be at. Great, thanks. So this afternoon, I would like to outline to you the prospects for a turning point in the development of nanotechnology. In the early days of the concept, the focus was on a long-term objective: flexible, programmable, atomically precise fabrication of intricate objects, including components for machines that could do a better job of assembly of intricate atomically precise structures. During that time, we have seen the slow maturation of the development of ways of making molecular structures that are adequate to get us to the first rung of the ladder. And in the last few years, a set of technologies has reached a threshold that will enable us to take a crucial step, and I would like to describe that step to you today. This presentation, this topic was first introduced last August and has been developing some momentum as a research goal since then. So the large transition ahead is from molecular self-assembly to molecular additive manufacturing. And I'll be hitting several topics here. One is where macroscale fabrication is today, the way we make things in the macroscopic world by systematic engineering. Atomically precise fabrication today, which is very different, very contrasting. Then how to build on that to develop a first generation of atomically precise molecular additive manufacturing systems. And then just a few words about early applications, which I will turn over to Sonia very promptly because she is the one who is working in the area where the greatest applications are likely to emerge. So first, macroscale fabrication. If you look around you, you'll see many artifacts. You'll see people, and who are not artifacts, but virtually everything else you see was made by machines or with the aid of machines. And those machines were made by machines or with the aid of machines. And tracing back the genealogy of machines, you'll find earlier and less automated and simpler machines. If you go back far enough down the family tree of industrial technologies, you'll find someone doing this kind of work: shaping materials in a direct way to make components and tools that could make better tools, more mechanized, more precise, that were used to make better and more complex tools, to make better and more complex products, and ultimately a very intricate technology base. Making complex objects has required complex machinery. Now, we'd like to be able to make complex objects in the molecular world to start climbing a ladder of technologies like the one from blacksmiths forward. But it would be very nice to not have to build very complex systems to make complex objects. And in fact, in recent macroscale industrial progress, we've seen a way of doing that. You have relatively simple machines, do X, Y, Z motion of something with respect to a platform on which one is building something. And the something that is moved takes some action that results in a little bit of material being added. Sometimes it's using laser to sinter a bit of powdered metal. Other times it's a nozzle that's putting down little bits of melted plastic, the most common form of 3D printer or additive manufacturing system. And the reason this is so attractive is that rather than having lathes which cut cylindrically symmetric objects or various other mechanisms for casting with special tools and so on, where many different machines are needed to make different shapes, instead, additive manufacturing, by adding small amounts of material at a time with X, Y, Z control, a bit of material here, a bit more there, can make a tremendous range of shapes, including today aerospace quality materials that are used. This is a component that Airbus is putting in aircraft that you may fly in, and was made by a 3D manufacturing technology, an additive manufacturing technology. Well, the essence of such systems, again, is some structural framework. Moving an X, Y, Z requires a structure to hold parts together, to hold a tool with respect to a workpiece, and stepper motors to increment on X and increment on Y and increment on Z to put the tool where it needs to be to add the next bit of material. So we have structural frameworks and stepper motors, and then some way of getting, in the typical case, materials to the location and then placing those bits of material. Not all 3D printers work that way, and what I will describe in the molecular world avoids that bit of complexity. So atomically precise fabrication today, back up and ask, how do we make complex things from molecules today? Well, the complex structures that are readily accessible today using chemistry, but also biology, because they're biomolecules first and foremost. One is DNA, which forms rod-like objects. The helix, the double helixes, can be thought of as a funny kind of twisted rod that can be used as a structural member. The other class of biopolymers that we can use to make structural objects and functional objects are proteins. My first paper in this area in the Proceedings of the US National Academy of Sciences was on the prospect of protein engineering. People thought it would be impossible because it was too complex to predict how a chain of amino acids would fold into a three-dimensional object. I pointed out that they were trying to predict how natural proteins would fold, and it's a very different task to design one that would fold the way you planned. And people said, aha, and that's the root of the citation tree. In this area where people have gone off and done brilliant work that is now coming to fruition in the direction that I had been looking for as a way of implementing the kind of system that I'm finally in a position to implement after all these years. So it's rather an exciting time. So the nice thing about proteins is that they're highly functional materials. They can be as solid as wood. They have shapes that can be tailored in great detail. They can serve as components for motors. They can serve as components for enzymes that serve as enzymes that transform other molecules and so on. So DNA is simple and soft. Proteins are a very good engineering material. The great advantage of DNA is, in fact, that it's simple. There is a field of technology known as structural DNA nanotechnology, which can now make 100 nanometer scale objects on a scale of, in terms of material content, millions of atoms. And if you follow design rules, making a new object is about as much of a research project as carpentry. There are ways of holding the helices together in a systematic way that all is based on Watson-Crick base pairing. And so these objects can be designed and made in a matter of days. Proteins have recently reached a threshold where engineering self-assembling protein objects, as of 2014, this report, reached the point of being able to systematically design complex objects. Here we have extended arrays, flat sheets, you can think of those as plates that could be used for structural purposes, work done in 2015. And the way this works is that you have to synthesize molecular chains using chemistry or biology. First, designing molecules that will self-assemble in solution. They have to have complementary surfaces. DNA, that's very simple, Watson-Crick base pairing. Proteins, it's a matter of designing pieces that will fold in a predictable way to make what amount to 3D puzzle pieces that will then fit together with other pieces to make a larger object. That's difficult, requires computational search. That's been a long process to get to the threshold that was indicated by the previous two slides. So this is very different from macroscale fabrication. You don't put the pieces where you want them to be. You do a very complex process that enables self-assembly, starting with one-dimensional chains. So what we'd like to be able to do is additive manufacturing, where you put the pieces where you want them to be. You can see that would be a fundamentally different way of manufacturing atomically precise structures on the nanoscale, enable faster, more systematic design, a wider range of materials, be a technology platform for a fundamentally new way of making intricate nanoscale objects with atomic precision. That is a very large prospect, and now finally there is a clear picture of how to get there. 3D printers require structural frameworks and steppers. It's still necessary. As for the tip functionality, want to simplify systems, you can substitute bulk fluid flow and diffusion to transport materials, and instead of putting pieces where you want them to be, you can take a workpiece and activate a location so that the building blocks that are washed in bind there. So three-dimensional designation of where the blocks go rather than moving the block, much simpler than what people had thought just a few years ago. Molecular chains are made to do by chemistry. You start with a block attached to a surface, activate a site chemically, wash in a block that reacts there, and then continue by activating at the end, repeating, and building a chain. And those are the kinds of chains that will then fold up to make objects if you're very clever and design such things. So the concept of 3D additive manufacturing with molecular building blocks is to have a tool, not a solution that washes through and activates the end of a chain, but a tool that can move around on a structure and activate a particular location. So moving to a particular place, activating the site, moving along to additional ones where you'd like to have the same next kind of block, wash in blocks of that kind, and then iterate additional cycles, additional layers, different kinds of blocks on each cycle to make an intricate three-dimensional object. This is a stack of bricks. There's no reason why the object has to be that simple in structure. So what's required for that are the machine and the building blocks and the tip for activating locations. If you have that, you can avoid folding chains to make puzzle pieces, have greater design freedom, wider range of parts, structures, and functions. Fast design and fabrication for product development, put the pieces where you want them to be. And a key point here is that if the structure, as I'll be describing, is based on self-assembly of DNA and proteins, for example, and some auxiliary molecules made by organic synthesis, you don't talk about how many devices you're making. You talk about how much material is being self-assembled to make those devices. And in chemistry, a milligram is considered to be a respectable but small amount of material, and a gram is considered to be modest but very substantial amount of material. Machines of this sort would be on a scale of 100 nanometers or so, and a milligram of material being, macromolecules assembling to make those machines provides 10 to the 12th at a shot. 1 gram, 10 to the 15th. So rather than having some large machine that's trying to do precise manipulation on a surface, instead the picture is of having a substantial volume, fluid flowing through carrying materials, 10 to the 12th to 10 to the 15th machines working in parallel to make things, to determine how those building blocks are arranged. Hence, scalable quantities, and eventually, by building arrays of machines, there's a pathway to scalable product size. Initial products would be on the scale of the machines, however, which is 100 nanometers or so. So the key components need building blocks, platforms to build on, activation tools. Those are questions of chemistry. There are many ways of solving those problems. None of them are obvious. Chemists do use very clever and indirect methods to make structures. What's needed here is clearly within the envelope of what chemists have done, but we're going to need a bunch of chemists to actually figure out what blocks they can synthesize that meet other system constraints, what the functional groups are that are activated by what kind of catalytic mechanism, and so on. The part that has been the sticking point, the hard part, has been the machine. And there, the two parts, again, are structural frameworks and steppers. So need a system that can move in X, Y, and Z. X and Y can be performed by strips of self-assembled structure that are in a framework something like this. Z-axis by moving the two U-shaped pieces with respect to one another, and each one is carrying the X slider and the other one the Y slider, and that provides the X, Y, Z motion. The reason that this is a boxy configuration is for structural rigidity. Biopolymers are relatively soft, relatively low modulus. All else equal, lower modulus means larger thermal fluctuations. Thermal fluctuations in a linear elastic model give a Gaussian probability density distribution for the position of the tip. And the thermal fluctuations are constrained by mechanical forces. The variance is inversely proportional to the mechanical stiffness. And if you have soft materials, you try to build a structure that is as rigid as possible with those materials, and one that I just outlined turns out to be a very, very attractive design in that respect. And then the next way of accommodating thermal fluctuations is to have the size of the blocks and the targets be far enough that the probability of hitting the wrong target is very low. So numerically, those constraints can be met in a part of design space that results in some very attractive systems. The other part, which is more interesting, and really some new ideas here, the structural mechanisms we're waiting for the fabrication techniques. What's new is a way of making stepper motors that seems very attractive. Probably other ways of doing it, but this one is nice. In the macroscopic world, there are what are called three-phase motors, stepper motors in this case, where turning on one set of windings produces a magnetic field that produces a potential energy function that aligns a moving part with respect to that. Turning that off while turning another one on shifts the location that the moving part wants to sit. You turn that one off, turn on another, it keeps shifting. You do the three in reverse order, moves the other direction. So each cycle moves a step. Because molecular actuators are at a premium, there's a trick where you leave out one of those steps and have it be kind of an idle step, and that gives three distinct states, which is enough to move left and right in increments. How to do that? Well, want to have three input channels for control. Well, a natural way to do control is by flooding the volume with light. If you can have absorption at three distinct wavelengths, you have three different addressing channels for your actuators. Turns out there are some dye molecules that absorb very nicely with well-separated peaks, modest amount of overlap. And by putting in light at three different wavelengths, you can activate different sets of molecules that are activating different phases of a stepper motor. What does that activation look like? Well, there are molecules of a family called azobenzenes, which when they absorb a photon, they flip around a double bond from a trans configuration on the left to a cis configuration on the right. That's enough to change the way they affect their environment, and if that changes the potential by kT, which is easy, that's good enough. Now, there are two problems when I was first thinking about this a few years ago. One is that these absorbed light only at very short wavelengths, the violet range. You couldn't channel multiple wavelengths of energy in there because you can only run the energy downhill to longer wavelength absorption. Well, in 2013, developed some clever people who were trying to get systems that would work in the body where red light travels more easily, figured out how to make molecules of this kind that switch with red light. The other problem was that the reverse switching, what took minutes to hours, that time has now been reduced to the microsecond or nanosecond range. Very recent developments, 2013, 2012. So we have the structures, due to recent advances in protein engineering and structural DNA nanotechnology. We now have components that are suitable for motors. So those are the key components. The key challenge is coordinated cross-disciplinary development. Almost all of this work, in fact, actually each of those technologies has been driven primarily by an interest in biological applications, biomedical applications. Researchers who are developing molecules for introduction into biology do not work together to build complex systems built from those molecules. It requires a systems engineering approach where there's a top-level function. You decompose the function into component elements, such as a framework versus a motor versus a control system, chemistry for building blocks. Those need to be put together. And last August, there was a meeting in Berkeley, meeting on integrated nanosystems for atomically precise manufacturing, which is exactly the direction that I've been pointing for some time. Person who this was held by the Advanced Manufacturing Office of the U.S. Department of Energy. The program manager is someone who I first propagandized when he was a doctoral student at MIT back in the 1980s. So another element that has come together is an organization that has a reputation for doing systems engineering. I guess it grew out of the Manhattan Project in part, and they also do renewable energy and many more peaceful developments. And they're now very seriously looking at developing this class of system. They are, however, moving at government speed. The workshop was in August. Apparently, the final workshop draft, well, they've assembled the material for comment by the workshop participants. I'm expecting to see it any day now, and sometime after that there will be a report from the workshop. I can report that there is also some preliminary interest around the Oxford community in addition or instead having a center of coordination for a systems development effort in this area in the Oxford milieu. We'll see where that goes, some very, very preliminary thinking there. So early applications. Well, discussed a lot of them at the workshop for DO8, many of them involving materials processing and strong materials and filtration membranes and electrocatalytic membranes for fuel cells, energy-related applications. But the most attractive applications, I think, are in diagnostic and therapeutic products in medicine. Our bodies are full of atomically precise structures, nanoscale structures. These structures interact with one another, which is how biology basically works. And to be able to make structures of that kind more rapidly, with more design flexibility, more control, and straightforward control of three-dimensional structure can be very important. To give some sense of the complexity of structures that people are investigating today with more conventional means of assembly, here are some nanoparticles. One class of applications of nanoparticles with tailored molecular components for nanomedicine. But at this point, I would like to turn the presentation over to a researcher in nanomedicine who can tell us what kinds of things are being built today that we could perhaps, by providing better tools, enable to move forward even more rapidly. Thank you.